All right, everyone. Hello. Welcome to Rearing Rambles, episode 5. Now that's what I call a good time. Let's all take a breath, sit back and relax. And I'm going to tell you about a couple of years ago, right when COVID first started in 2020, my adventures pollinating the pink lady slipper, Cypripedium acaule. So let's talk a little bit first about Cypripedium acaule, shall we? Cypripedium acaule is the pink lady slipper, and it is one of the more widespread lady slippers across the United States. The genus Cypripedium contains about 60 species of temperate and subtropical orchids that have very large, inflated front petals that almost make them look like slippers, which is why they're called lady slippers. Um, Cypripedium has numerous species throughout the United States, as well as Canada, as well as Asia, Europe, and I believe it's restricted to the Northern Hemisphere. Now, Cypripedium acaule was the really only species that I would ever encounter in Connecticut and Rhode Island. This lady slipper has some unique features to it. Unlike a lot of the other Cypripediums, this plant does not produce a leafy stalk. Rather, it has a pair of leaves at the base of the plant, from which, out of the center, a flower stalk emerges. In May and June, the scrotum-like pink flowers can be seen throughout the woods in patches. This is a species that needs very acidic soils to thrive, and it's very commonly found near pine trees. Unfortunately, because of how attractive this particular plant seems to be, people like to pick it, people like to dig it up and poach it, and in some areas it has become quite rare as a result. Now, it hasn't really gotten much attention as far as listings on any Endangered Species Act or anything like that. However, remember that basically all extinct species were once very common species. Cypripedium acaule has a somewhat unique ability to remain dormant for up to 7 years during adverse conditions. And so doing population censuses on this species can be somewhat difficult because on a given year you may not have every mature individual of the population actively growing. Some work that has been done on this species has indicated that there is a fairly narrow range of genotypes represented in any given population, which indicates a high level of inbreeding and a high level of resistance to the deleterious effects of inbreeding. Now, I know that I've been promising a podcast episode specifically about inbreeding. Maybe that will be next episode. But Cypripedium acaule doesn't seem to have any problems with, selfing or cross-pollinating with a very close relative. Now, we need to talk a little bit about orchid biology here. For the folks who are unaware, basically, all but just a handful of members of the orchid family have seeds that require a symbiotic fungus in order to germinate. The seed has to land on this fungus, and the fungus has to actually grow into the seed and provide that embryo with nutrients and energy if that embryo is going to grow. Now, in culture, you can use a sugar-based medium that has various compounds in it, such as vitamins and other things for that seed to actually germinate aseptically in the absence of that fungus. And we'll get to that a little bit more later in this podcast episode. But that's going to be important because Lady slipper populations have diminished across the range, and there have been some efforts to, for conservation reasons, propagate lady slipper orchids. Lady slipper orchids provide a unique challenge, however. Unlike most orchid species, the tropical ones, that seeds can simply be removed from a green, immature seed pod, placed atop the surface of a generic plant medium, or a plant medium that's slightly more dilute with a little bit of extra charcoal, because that's generally what orchid germination medium is. And then they will germinate. Lady slipper seeds have dormancy factors imbued in them fairly early on in their development that require some extra effort to break. But we're going to get back to the idea. So pink lady slippers are pretty common, but like all the lady slippers, they are declining in number, unfortunately. You only see this lady slipper growing in places where the fungus that the seed relies on for germination is found. Again, it's very often associated with pine, but not always. It is always associated with very low pH soils, however, in well-drained spots. Another interesting thing about Cypripedium acaule is it's an orchid that you can see a great deal of phenotypic plasticity. Phenotypic plasticity is this feature of an organism where it has the ability to change its appearance in order to match its environment. And so Cypripedium acaule that are growing in dense shade have much broader, darker leaves to capture as much sunlight as is possible. Whereas when they're growing in full blaring sun, the leaves are very small, very bright colored, reflecting a lot of light, losing as little water as possible, because the same photosynthetic output can be achieved with much less leaf surface area. So Cypripedium are orchids that are fun for me because they present a little bit of a challenge. They're not typical in that you can just sow the seeds and get them to germinate. Now let's talk a little bit about the acaule populations where I am from. So we have a couple of wildlife management areas in Rhode Island where I like to look at the lady slipper orchids. In Massachusetts, there's Douglas State Forest that is also full of the lady slippers. Very fun to observe them during the last couple weeks of May, first couple weeks of June. The exact flowering time is going to depend on things like how quickly the ground thaws and how warm of a spring we have and things like that. They're not found all the way out into Colorado. It's just too arid here. The soil pH is way too high for this species. We do have a Cypripedium in Colorado, but it's not acaule. And I am not the biggest fan of Cypripedium acaule, if I'm honest. I think they're funny because the flower looks like a scrotum hanging off of a stick. And that does have an appeal to it. But I do really like Cypripedium parviflorum better. I like Cypripedium macranthos better. I like Cypripedium candidum and Kentuckiense better than Cypripedium acaule. Not that I hate Cypripedium acaule. It is a nice plant, but it is not my favorite of the Cypripedium species. But that doesn't mean that I'm going to not grow it. And there are a couple of reasons why I want to grow it. I have some friends who want to grow this particular lady slipper compared to the other members of the genus. And it does have some features to it as far as its behavior in vitro or in tissue culture that make it a little bit more of an enigma, we should say. Not to the extent that Cypripedium californicum is an enigma, but it does get kind of close. Now, Cypripedium acaule um, is found in various areas throughout the United States, especially where I'm from. And it gave me a good opportunity to practice kind of just the processes of collecting and sterilizing and sowing native orchid seeds. Now, when I do my episode on Pogonia ophioglossoides, we'll talk a little bit more about some challenges with getting temperate orchids to germinate. But for now, we're going to stick to Cypripedium acaule. Now, Cypripedium acaule is a nice plant to work with because it was convenient. It was there. And so let's talk about my conversations with somebody at the University of Rhode Island. The two of us were wondering if Cypripedium acaule, well, it was actually conversations with two different people. We were wondering if we don't see very many seed pods on the Cypripedium acaule plants in the fall because of pollinator limitation or because of predation of the seed pods by deer, chipmunks, and other things. And so I set up a little bit of a small experiment that I'm going to get into in a moment. But Cypripedium acaule is thought to be pollinated by bumblebees that, accidentally end up in the flowers and crawl out pollinating the flower as it passes, through the front pouch of the flower. Now, whether or not that's actually how they get pollinated, I'm not quite sure, but we'll just go with it. Now, it would be very concerning from a pollinator perspective if bumblebees were in such serious decline, which they are, that they are not doing a lot of Cypripedium acaule pollination because there just simply aren't that many of them out there. With some pollination syndromes, there's a minimum effective population of your pollinator, and so Cypripedium acaule may be screwed in the long term if we keep losing our bumblebees. And so, in 2020, and we were really cutting it close because Governor Gina Raimondo in Rhode Island had shut down the wildlife management areas for quite some time, right before the acaule were coming into bloom. Now, unfortunately, the acaule populations that I'm going to talk about are no longer there, and I'll talk about that later. But my friends and I, they reopened the wildlife management areas right before the acaule were actually in bloom, which is good. And we went around to two separate areas, Big River Management Area and Carolina Management Area in Rhode Island, and pollinated dozens of these pink lady slippers. Then I set cages, little, like, maybe like 1 foot tall by 6-inch wide chicken wire cages around the plants and anchored them to the ground. Now, these accommodated the entire plants, and the goal there was to exclude anything that could have eaten those seed pods, okay? Sounds simple enough. So we left, and then September rolled around. When September rolled around, I went back to those areas, and here's what I found. Not only did, there were a couple of cages that were pulled up and taken away, but not only did basically all the plants within those cages that were left have seed pods, there were seed pods on a lot of the plants that we had pollinated, which tells me that Cypripedium acaule is not limited by things eating the seed pods, but rather limited by pollinators. There just aren't a lot of pollinators around pollinating these things, causing a low number of seed pods produced per year in the wild populations. Now, this isn't that big of a deal for an individual plant because they live over 150 years, but you can see where this might cause a problem long term over many generations of this plant. So, I had a lot of fun going around pollinating all these lady slippers and then going ahead and collecting the seeds. Now, Cypripedium have some unique challenges when it comes to getting to germinate. And I'm not going to go through the entirety of the protocol, but basically there is a germination medium that you make for Cypripedium orchid seeds. It contains a number of microelements as well as glucose, agar, water, obviously, and, vitamins, things like that. You make your material, okay? Make your germination medium. Depending on the species, you add a little or a lot of potato to that medium. Russet potato. Some species, like Cypripedium reginae, need less than, I think it was like 8 or 9 grams of potato per liter of medium, whereas other species like Parviflorum need closer to 196 grams per liter of potato. So it varies by species. Acaule is on the lower end, requiring, I believe, like 16 grams per liter or something like that of russet potato per liter of germination medium. Whatever. So it also requires BAP or kinetin. Those are a plant growth regulator that helps to hasten germination. So you make up your acaule medium, you autoclave it. You either pour it in plates, you put it in slants. You do what you do with it. I now pour plates for my Cypripediums. I used to do slants in borosilicate tubes, wherein I would take the tube and put 25 milliliters of medium and the appropriate amount of potato for that 25 milliliters of medium. Autoclave that and just streak the seeds across the surface of that. But I found that using petri dishes is just a lot more convenient now that I have my own laminar flow hood. It's just a more streamlined process. So I make my medium, I blend it up really well to get that potato incorporated. I put it in bottles and autoclave that, and then I pour it on the plates. And then I streak the seeds across the plates after their treatment. So how do you pre-treat the seeds of Cypripedium and other orchids? The first step, if you're using mature seeds, is to use vacuum pressure and some tween water. Tween is a surfactant or a wetting agent. It helps water permeate things that it otherwise wouldn't be able to. You put the seeds in some tween water. You apply vacuum pressure until the seeds sink so they've taken up water into their coats. And then you bleach the seeds. Some species of Cypripedium require bleaching for about 10 minutes, whereas other species require several hours of bleaching. And 10% bleach, about 0.5% sodium hypochlorite. And that does a couple of things. One, it sterilizes the seed. Remember, you're making an agar that's loaded with vitamins, sugars, minerals, things that bacteria and fungi really love. And because bacteria and fungi will snuff out your orchid seeds, you have to keep that medium sterile. A contaminated medium is no good anymore. When I do tissue culture, I do get contamination. Depending on what I'm doing, it can be as little as 5% or it can be as high as, 60%. It depends on just a number of factors. And, for those of you who want to say, what do you mean you're getting 60% contamination? Please keep in mind that I don't do tissue culture in a dedicated tissue culture space, and it's not not the only thing I do all day. If I was working in a professional tissue culture lab setting where, all of my cultures are isolated from any other, living organism like plants in soil or fruit flies with mites and other things, then, 60% contamination would be pretty bashful. But I think given the fact that I live in a house with 2 dogs and 2 other people and however many insects and plants within pots full of soil and all those microbes, I think that, when I get, even 10% contamination rates, it's, to be expected. So anyway, you gotta keep the, seeds and the medium sterile. So you sterilize the seeds with bleach. The other thing that bleaching does is it removes the dormancy factors such as abscisic acid and some other things from that seed so that it can germinate. And so bleach your seeds. Acaule requires an hour-long bleaching time. Now, what I will tell you is that our Rhode Island population seems to need longer bleaching than the Minnesota population. And my hypothesis about that is the fact that in the fall of Minnesota, it gets really, really cold really, really soon. Whereas in Rhode Island, it takes a little longer to get cold. And cold breaks down those dormancy factors in the seed. And then the cold itself stifles that seed germination. So plants in warmer climates where the seed has to sit around and wait a little bit longer for it to actually get cold are going to need more germination inhibitors in order to sit out the rest of that season before it gets cold. So I collected the seeds from the plants, and I did a little experiment with some of these seeds. I collected multiple seed pods. I left a lot of the seed pods intact on the plants in the wild populations because I figured the populations could use a good boost of, fresh seeds that year, whatever. I had taken those seeds and I did an experiment with the potato. I made potato powder and added it to medium as a dry weight equivalent of the amount of fresh potato that I would need per liter of medium. So you need a certain amount of medium for the fresh potato for this species. The dry mass equivalent is basically, whatever minus the water weight of that potato mass. And so I did an experiment where I went ahead and took the acaule seeds and put them on germination medium with either dry white potato equivalent or regular fresh potato. And I didn't find any statistically significant difference in germination rates between the two groups. I may run something like that again. I am running an experiment with Cypripedium reginae, which, I might talk about in just a minute. We can talk about Cypripediums in this episode. I can talk about Cypripedium for quite a while. So, that's an indication that you don't need to use fresh russet potato. And I know what you're thinking, well, what does it matter? Well, you can actually make medium premixes customized to different Cypripedium species by making the powder potato, putting it in a medium powder, and then sending it off. It takes up less space to store. It stores longer than fresh russet potato. And there are fewer phytosafety and sanitary concerns with dry potato powder than, actual fresh, wet russet potatoes. So the acaule seeds will bleach for about an hour. You rinse them, you put them on your medium. Cypripedium seeds need to be germinated in the dark. So light inhibits their germination and development. In the wild, they will be underground, engulfed in fungus. They need dark. So you start your Cypripedium seeds in the dark, and the seeds will germinate after several weeks. Cypripedium acaule takes usually between 8 and 10 weeks to start seeing germination. Although I got a batch this past year from people in Rhode Island, and they took about 6.5 to 7 weeks to germinate, which is unusually fast, and it was a very high germination rate as well, which really did surprise me. And so I had these acaule seeds on both medium that contained potato powder and raw potato. They did just as well. Then I replated them. Now, unfortunately, this was about the time that I then moved across the country from Rhode Island to Colorado, and I lost basically all the acaule to contamination. Tissue culture stuff doesn't move or ship very well, and so a lot of stuff that I had done the replating of contaminated during my move, which was unfortunate. So anyway, the past year then, I worked on getting some new acaule. I did lose some Cypripediums to some other things that happened. I had there was an autoclave failure, and the reason I know it was an autoclave failure is because I had jars of replate medium. So basically, the seeds they germinate, they get to a certain size, and you move them to bigger containers of medium, and you plant them more sparsely so they can grow into seedlings rather than just protocorms. And I had a lot of jars of replate medium that didn't even get opened and put anything on, contaminate, and that's usually a sign that the autoclave in one of the buildings at CSU is perpetually broken. There's always something wrong with it, so I don't use that autoclave anymore because I've lost too many batches of medium throwing it through that autoclave, and then you know things contaminating after the fact, and I just assumed that there was something that the autoclave didn't heat up enough or whatever happened. So that's always something to be aware of when you're doing tissue culture stuff. So, um, I do want to shift gears a little bit today. Um, we will come back to Cypripedium because this really should be about Cypripedium acaule. But I also wanted to talk about some other Cypripediums. Because I don't just grow Cypripedium acaule, I grow a number of Cypripedium species. And right now I'm doing an experiment with Cypripedium reginae. So let me give a little bit of background. This is the first year that I'm really doing a lot of serious work with plating up my seeds rather than putting them in slants. And because of that, I sowed a good number of extra Cypripedium reginae seeds. Big mistake, because I have over 4,500 protocorms of Cypripedium reginae. Not only did I lose very few of the Petri plates to contamination this year, I also had a lot, very high, 85+% germination rate. And as you can imagine, because each seedling needs 10 milliliters of replate medium to complete its development, 45 liters of replate medium is not a trivial matter. And Cypripedium reginae is not the only species that I'm working with. And so I can't, in all good consciousness, allocate all of my time, energy, and resources to one Cypripedium species. Now, Cypripedium reginae happens to be one of the more quick Cypripediums to develop, so I have plenty of time to replate the reginae before I have to get on to replating the other species, fortunately. But what it basically means is that I have this backlog of reginae protocorms to deal with. So I am doing an experiment, and that experiment is testing different protein sources on the development of the protocorms. So, Cypripedium need a source of nitrogen in order to grow. And generally, that is given in the form of a protein. In the Cypripedium germination and replate media, you use casein hydrolysate, which is basically an enzyme-digested casein, which casein is a milk protein. Now, there are other sources of protein, though. And milk protein is a lot more expensive and a lot less sustainable than, I don't know, insect protein. And if you don't know where I'm going with this, I got some dried black soldier fly larvae, and then I dried some dead lobster roaches and crickets, ground them up, and so I have a couple of liters of the Cypripedium reginae replate medium with the protein equivalent in black soldier fly, lobster roach, or banded cricket powder. And I'm going to replate the protocorms on these different media. And I'm going to see how they develop. I'm going to see if I can use a replate medium with insect protein to successfully develop the seedlings of a Cypripedium. And if I can, then it's possible to use that as a substitute for milk protein. And it would save people a lot of money. And it would be more sustainable for the environment. Because we like the environment around here. So, we're about halfway through my time allotment for this podcast. I do apologize that this is getting out a little bit late. I've had a really busy semester, and while I try to get everything uploaded by 10 a.m. on Fridays, it just didn't happen this week. I will probably, if I have time before next Friday, pre-record another episode or two so I can just schedule them to upload at the normal time. It's just been a little bit of a challenging couple of weeks. And so while I try to get things out on time, I have, 11 other things going on any given day. And, um, this isn't my primary focus. But I do like to ramble about my experiences rearing things and growing stuff. So let's see what else. Now, Cypripedium acaule, is an orchid species that is a little bit slower to grow than the other Cypripedium. And again, it likes a low pH soil. Man, I'm tired. In case you've been watching the video portion of this, I started recording last night, and now it's, noon on Friday when I'm recording the next bit of this. I was just too tired to really go on with last night. And I'm trying my best. And so, let's talk a little bit about some of my goals with Cypripedium propagation. Now, I like to propagate orchids, and I like to grow a lot of things, especially things that are challenging, and Cypripedium can be fairly challenging. I'm not afraid of challenges, and I'm kind of stubborn. I am trying to help some people with their plants, and I've reached out to some people who are trying to do their own propagation of these plants, and I've offered free seeds and free stock solutions and things like that. And the reason why is because I started by getting free seeds from people, and, a lot of us in the Cypripedium community aren't necessarily looking to get rich. We don't want to grow these plants so we can make a ton of money. We want to grow these plants because we want people to appreciate them, we want people to obtain them through more ethical means, and we want to improve the populations of these plants by giving them a little bit of extra help. Now, I don't have any, I have one acaule seedling right now, but I don't have any seedlings that I've grown for a year of Cypripedium acaule, but I do have other species that I have some experience with. So let's talk a little bit about the cycle here. So when you grow Cypripedium seeds, you get the seeds in, the fall, October, November. You store them in the freezer until you're ready to use them. You pre-treat them and you bleach them after you make the medium. The medium composition is basically, let me pull up my stock solution. So I will read off what you want to do here. Once I find, let's see, dang it, I believe, here it is. Here, let's see. Wait, that's not the right, just a second. Is it this? Sorry, I have so many different Excel files. Dang it. No, medium composition. Here we go. Okay. So, I'm going to read off my ingredients, and I'm going to tell you what I do. So, I make a stock solution that has, 100 grams of potassium chloride, 200 grams of potassium nitrate, 200 grams of potassium phosphate, 19 grams of ammonium citrate, half a gram of boric acid, 0.025 grams cupric sulfate pentahydrate, 25 grams ferric ammonium sulfate, 97.69 grams of magnesium sulfate, 1.54 grams of manganese sulfate, 0.02 grams molybdic acid, 0.1 grams of potassium iodide, 0.5 grams zinc sulfate heptahydrate, and 0.1 grams of myo-inositol. I dissolve that in 3 liters of water. So every liter of medium will have 3 milliliters of stock solution added to the liter of water. And then you're going to add between 10 and 20 grams of glucose. Most species use 20 grams, but there are some species like Cypripedium kentuckiense where you're only adding 15 grams of glucose, and species such as Cypripedium calceolus, which you only add 10 grams of glucose. 9 grams of agar. Cypripedium kentuckiense, you put 0.6 grams of casein hydrolysate. For everybody else, you put 0.4 grams. Same thing with calcium nitrate. You blend that all together. And then some species you add a lot of potato. Some species you add very little potato. So, for example, you have media where it's only 23.2 grams of potato per liter, and then some where it's 185 grams per liter potato. Sometimes you add BAP to the medium, sometimes you don't. It just depends on the species. And in general, you add between 0.3 and 0.5 grams of casein hydrolysate and calcium nitrate. You make your medium, and you autoclave it and everything else. Then you will pour it out, and so I do this in November. And then I get all my seeds, and I vacuum treat them, and I bleach them, I sow them, then I put them in dark boxes and ignore them for about 6 weeks. 6 weeks pass, and I go and I check. And I throw away anything that's contaminated. Usually I get 10 to 40% contamination. Some species seem to contaminate a little more easily than others. Again, it's not perfect. If I were working specifically on just tissue culture stuff and I had a dedicated tissue culture space, I would see, probably lower contamination, but I don't have that luxury. And that's okay. I've gotten a lot better with my technique, and so I've gotten a lot lower contaminations over the years, which has been great. I mean, I'm still producing, thousands of protoforms and hundreds of seedlings. I don't need everything to survive, honestly. It would be a little bit more tricky if everything made it, but that's a different matter. Species such as Cypripedium reginae, Cypripedium parviflorum will be germinated in a Cypripedium guttatum. They'll germinate within about 6 weeks. Guttatum might take closer to 8 or 9 weeks, but you'll start to see some activity as early as 5 or 6 weeks. Then I ignore the acaule for 2 months, and then I repeat that. I do that process of just going through and throwing away anything that's contaminated, leaving the stuff that's not. Cypripedium reginae needs about 2 or 3 months before those protoforms are big enough to replate. They'll be about the size of a quinoa grain, and they'll have a visible nub at one end. That's the bud that'll eventually become the shoot. And then a growing root apex that kind of looks like a little green tip on the other end. They're kind of spear-shaped. It's really cool. Don't worry if some of the protoforms are a little bit of a different shape than others. There's going to be some variation. You're going to have some that are more boomerang shaped. You're going to have some that are more rotund. They'll all still grow into seedlings in general. I do occasionally get these weird things where it just looks like a big wad of whatever, and it doesn't actually grow, it just dies. That does happen, but generally the protoforms can't be quite variable and be just fine. Then I make my replate medium. The replate medium uses the same stock solution, but a little bit more of the casein or other protein source, no BAP, and less potato in general. Everything gets, I believe, crud, what was the, I don't remember the, exact the exact amount of potato per liter. I'm not going to check it out. It doesn't really matter. But you can reach out to me and we can discuss it more in depth later. I make the replay medium and I autoclave it. And the replay medium is not in plates, it's in containers. I have two types of containers that I use. One is jars, glass jars that hold 150 milliliters of medium, so I can put 15 protocorms on. And then I have these polypropylene containers that I really, really love. They're from Berlin Packaging. They're these square food containers. Make sure it's all polypropylene, the lid and the container, because polypropylene is autoclavable, polyethylene is not. Polypropylene is what you want. It has to be polypropylene or glass. Okay? Polypropylene. Polypropylene. And they're ventilated, so there's some airflow. I use a filter disc that keeps out contaminants from the air but allows oxygen in, carbon dioxide out. These plants are not photosynthetic yet. They need oxygen to undergo cellular respiration. They're not going to be photosynthetic until after their first winter. Please remember that. So, February, March, April is generally when I'm replating things. It might not be until June or July that the acaule get replated because they're more slow to grow, but most other stuff is replated by about late April. That way, the seedlings are fully grown by the end of the season, October, November-ish. Then they can be plucked out of the medium, rinsed carefully, placed in bags, freezer bags, and placed in the refrigerator for the winter. The first 2 winters they can't freeze. These seedlings are very delicate right now. The first winter after they've been deflasked, they have a dormant bud and roots. They're cleaned off, kept slightly moist in Ziploc bags, and kept in the refrigerator, away from freezing temperatures. In the spring, they're planted into an appropriate potting medium. They're put under lights and allowed to grow. Now, here's what trips people up. The first year, they're going to put out a couple of leaves. They're going to still stay very small. They don't need a lot of fertilizer. They don't need a lot of light, but I keep them under a grow light. They're going to go dormant pretty early. They go to bed early. They're little kids. They're babies. Don't panic. If the plant is slowly turning yellow and dying back to the ground, it's probably just going dormant, okay? Even if it's July, I've seen them go dormant as early as July, they're just going dormant. They're going to bed. Just let them be. When it comes like late October, put them in the fridge, things like that. And then the next year, they will probably go to bed a little bit later. And you'll see that trend continue. So, I wanted to put that out there for folks who are interested in growing Cypripediums. They provide a good challenge. So, back to my interesting batch. So, I ran out of seeds of the acaule that I had done my pollination work on, and one of my people back in Rhode Island has acaule growing out of her yard. So, interestingly enough, she had several seed pods on these plants. They must just be growing in a really good location because they're always producing seed pods. There aren't a ton of them. There are just a few, but they're always producing seed pods. And so she collected seed pods and sent them my way. I took the seeds out, storing them, and what I noted was that not only was there very high germination rate, they took a lot less time than normal to germinate. So I call her up. And I say, can you tell me about last year in Rhode Island? Was it a spectacular year for plants? Because the better the plant is doing, the better the seed quality is going to be generally. And what she told me was really weird. It was a very hot, dry summer last year in Rhode Island. And I was a little surprised because, my friends in Minnesota have the opposite thing happen where when it's really hot and dry in Minnesota, the plants don't do very well and the seeds are poor quality. Especially like Parviflorum and Californicum seeds, they're really impacted by bad weather. To the point where you shouldn't bother sowing seeds from a really dry year. And I was told, no, it was pretty hot and dry in Rhode Island. And I don't know if maybe maybe these plants were growing in a more sheltered location than normal, and so they, maybe got some extra water. Maybe the fungus they were associating with in the ground was, protecting them from the effects of the drought. I don't really know. Something was going on. But, those seeds were good quality. The other possibility is she may have harvested them a little earlier than I would have, which may have had an effect on their rate of germination and things like that. I don't know. What we do know with some Cypripedium species is that younger seeds, before the pod is fully developed, have a higher germination rate than older seeds, more mature seeds. So that's a possibility, but they're storing like mature seeds. They're not like losing their viability very quickly after storage. So they were mature enough, but I don't really know what was going on there. And so there are still a lot of unknowns. So yeah. Going forward, I would really like to get some more stuff done with the acaule. I have a friend in Rhode Island who is interested in these plants, and I want to send him some of these seedlings that I get once they're big enough. And I have some people who really helped me back at URI with some of my tissue culture stuff that I want to send some plants off to as a thank you. So I'm going to work on that. And, you know, one of the things is that the people who really pioneered a lot of the early work in germinating out these orchids, they're old. They're at the end of their life cycle, their lifespan, and I'd really like to just kind of continue with some of these efforts because um, there are a couple of things about this that are important to me. One is still having that connection to natural history, plants, and, the process of generating these organisms. It's not about having a large rearing or breeding operation where I'm making money and, being a kingpin of, orchid growing. It's more about fostering the interest, fostering the passion for this process and for this plant, fostering the concern about the plant's long-term well-being, fostering methods for getting the what's it called, conservation work done. There are people that will grow native orchids because they want to plant them out in areas that have been disturbed and, where they used to live and things like that. Getting, the plants propagated in a manner that reduces the need for people to go out and dig these up. Getting more people involved because it's good for the plants' wild populations and it's good for people's appreciation of the plants. And a lot of these people who work on these orchids um, are like that. We don't want to keep our protocols secret. We don't want to hide how we get this stuff done because we want as many people to be involved in doing this as possible. Because here's the thing. I put out a protocol. A lot of people put out the protocols. Maybe 0.5% of the people who read that protocol will actually follow it and successfully do it. Most people are still going to want to just buy the pre-grown seedlings. For me, the challenge of teaching myself and learning, and I'm not saying this is all self-taught. There are a lot of things that other people have taught me along the way, and I'm grateful for that. But you know, it's tangential to what I do as a career. I don't have a career in tissue culture. I don't have a career in orchid conservation. I don't have any of that. My career research is in insect nutritional ecology, plant-insect interactions, and things like that. I'm not, by trade, an orchid propagator. But the challenge of getting these things to germinate and grow into seedlings, rather than taking the easy route of just buying pre-grown, pre-hardened seedlings that are still fragile, still very delicate, but not as delicate as a protocorm, is really what makes me tick. And, I think that's kind of the logic behind sending out the free seeds. Um, so the people that send me free seeds, um, these aren't seeds that they would have otherwise grown. They collect plenty for themselves. And the rest of the seeds would either just be left on the plant to disperse in nature or just be stored in a freezer for years and years. Um, and that's kind of, the part that you want to make money off of is the mature seedling, because you've actually spent money on all the ingredients for your medium and the time plating up and replating and things like that. It's not about, you know, selling the seeds. It's about, you know, if somebody has the capacity to propagate the seed, let them, you know. And I want to make this sort of thing accessible to everybody because it's fun, it's cool. Um, it's not something everybody can do, but the other reason why it's very exciting to me is because it's a different life cycle. Orchids have a different way of growing from seeds than other angiosperms, okay, and other seed plants. Orchids do a different thing, and it makes them successful in the sense that they're speciose. It's the largest plant family. We can get into the nitty-gritties. Is Orchidaceae larger than Asteraceae? We don't really know. Um, but it is a successful, if we want to define success solely as speciousness, which I don't think we should. I think that it's one measure of success that's very specific when you're talking about, like, cladistics, but I don't think it's the only measure of success. Um, there are certainly problems with orchids being so hyper-specialized to require, and orchids aren't the only angiosperms whose seeds are like this. There are some other groups, certain Ericaceae, for example, where the seeds also require mycorrhizal symbiont in order to germinate. We talked a couple weeks ago about the Lycopodiaceae that are like this with their spores. I think fungal symbiosis is really cool. Maybe I'll do an episode on fungi. Um, I'll probably do that in lieu of an animal episode, just because fungi are more closely related to animals than plants. But I'm not sure yet. So, I don't have much more to say. But I will leave you with a couple of things. Please, please, please do not dig up wild orchids. Unless it's something like Pagonia, where, like, you take a piece of the rhizome because every year the rhizome grows, like, 30 feet across a wet meadow and it's not going to harm the population to take a couple of cuttings from it. Don't dig up wild orchids. Be ethical. There are plenty of people offering seed-grown native orchids. If you're ordering orchid seeds, please request that the seller put the seeds in a rigid container so they don't get crushed during shipping. I've had that happen. Talk to people who have grown orchids from seeds before. Most people are very willing, when it comes to native wild orchids, to share their propagation protocols. I find that orchid folks, especially some of the people working on the native temperate orchids, are very friendly, very cooperative, very nice people to deal with. I highly recommend having conversations because, some of these people aren't going to be around for much longer. And it's sad. I'm not always that attached to people, but when it comes to some of this sort of stuff, it does make me think. And it makes me be very grateful for some of the people I've come to know through just wanting to grow orchids. I started the Cypripedium stuff because back in Rhode Island, before I went out and pollinated all the acaule, the year before, I collected a seed pod of acaule when I was just out hiking, and I put the seeds on some medium. They didn't really germinate because I didn't know any better. I didn't find online protocols until after. I had from Phytotech Labs, the Cypripedium germination medium, but I just made it up as it was. I didn't add potato, I didn't add BAP, I didn't bleach the seeds, I bleached the seed pod, and, I really screwed some of it up because I just didn't know any better. But I was interested enough where I was kind of like, I want to just germinate all sorts of orchids for for fun. And then I got a hold of somebody who was doing it, more seriously than I was. And that's how I started. And, the first couple of years went really, really poorly. Um, for a number of reasons. Part of it was I was a little stubborn. I was still young, I was like 19. And I don't want to say I didn't appreciate the advice from other people, and it's not that I thought I knew more. I just, I thought that I had a better sense of necessary versus unnecessary steps in something than I actually did. And that's an important thing to acknowledge. Um, I do recommend when you're given a protocol, trying to simplify it for yourself. But before you go ahead trying to simplify it, make sure you have it down pat the way that somebody else has it down pat. Okay? Once you have a protocol down pat, then you can try to see, are there steps that I can forego? Don't do that beforehand like I did, because you'll end up getting frustrated. I was frustrated but not discouraged, and now I am able to get a lot more done because I'm more willing to listen. You know, and I don't want to say it was. I don't know if I would say it was arrogance. I would say it was more just me being a naive, late teenager who, you know, figured that I could get away with things that I didn't understand the underlying mechanisms of. And, I'm constantly improving my techniques. I'm constantly, looking for new ways of doing things. But I do advise people to not be so quick to try and reinvent the wheel. That, because you got to think that some of these things have been tried and failed before. So yeah, thank you for listening. Thank you to everybody who's been essential in my journey with this sort of stuff, and I hope to catch you all in the next one.